So, I mean, electric signals are interesting because they are life signals, and we are trying to understand like different aspects of life, right? And they are interesting because they tell us something about the structure that is generating these signals. And I think you will agree with me that basically anything that a biologist or a doctor or a psychologist does is somehow trying to extract these signals that will allow us to understand like the organisms that are producing the outputs that are related to life. Like, for example, we know that the heart rate is an important signal that we can measure in life. And the heart rate is going to change a lot across the evolutionary spectrum, and it's going to tell us things about the function of the heart and about the architecture of the body in which this heart like is pumping blood, right? We know that like higher metabolism animals will have higher heart rates. We know that the smaller animals, they can have like higher rates because they can allow themselves to have higher firing rates. I was going to say like higher heart rates because their bodies are small, so very fast they can like change this blood supply across the whole body and everything. So from the heart rate signal, we can extract a lot of information about the structure, among other things, because the heart rate itself is gonna be like totally depending of the structure that is being producing the heart rate, right? It's going to be related to the like size of the animal and the volume of the animal. And this is what we have a relationship between these like variables, right? We have this relationship between heart rate and mass, and this is something that this allometry is something that we will find across many of these signals. Like going back to the main concept, the idea is that we have like a strong relationship between architecture and signals, and this is why it's interesting for us to study signals, because it's an indirect way but very powerful way to study like this the architecture of life, right? This is, for example, the same thing that happened with the violin family, right? Like we know that they will different like favorite like resonant frequencies, and this is going to change as a function of the size of the violin or any instrument, right? And this is just like basic physics. Okay, like the interesting part with the brain signal is that they are totally off with this. In this case, and this is one of the few or the only exceptions that we can think about, we find like the opposite relationship. Now is the architecture of the brain what is subordinated to its own signal, meaning that studying brain signal is very important because the brain had to keep the signal as they are because they have their own meaning. Like when we are studying some oscillation of some given frequency, the brain has evolved as it changes size, it changes shape, it creates like new cell types, whatever, to try to keep the oscillations at the same frequency. Because if the frequency changes, the signal changes, then the function is going to change. So then here studying signal, it becomes even more powerful because understanding the intrinsic properties of the signal itself, it will allows us, it's like a window to understand like the operation of the brain itself. I hope that the concept is more or less clear, right? Like allometry in life rhythms is something that we have studied in the school. And like the beauty here is that the brain is breaking this rule and we have like the opposite relationship. We have here like a subordination of the brain signal to the architectural aspects, okay? Okay, and then we go directly to the electric brain signal. So here I was just trying to frame why we think it's fascinating to study electric brain signals. So the most simple electric brain signal that we can think about, and one of the earliest that was studied, was like the action potentials. If we remember, like the squid experiment, right, that got the Nobel Prize and everything, the Huxley and everything. So we know that most of what is happening in our brain, on most of what we experience, can be like in the last extent, like resolved at the level of action potential, right? Like anything we feel, think, and experience at some extent are action potentials in the brain, and we can study and we can see them, and we have studied also in the school, right? Like the neuron, they live at some membrane potential, and at some point they cross a threshold and then depolarize because they active like sodium channels that are voltage dependent, and then these channels are gonna depolarize the neuron, and this will activate like channels that are now like potassium channels that would be open like a higher like voltage, and this will repolarize the neuron, and then like there is some mechanism that keep the neuron potential at some millivolts. So I guess like most of us, of all of us, like we are familiar with the concept of an action potential. Okay, so then this is the concept of allometry I was saying. Like we find a like super strong relationship between architecture and like life signal, almost anything we are looking for. And the same happen with almost like anything like we can measure like out in the nature. And then brain signal, they are an exception, and they show this totally flat relationship between the architectural aspects like of the oscillation, which is the brain size, and the frequency. And this happens because here the brains are breaking this rule, because the frequency itself will have like important function, as we will see today. Okay, I hope that now is a little bit more clear. Okay.
Yes.
Thank you. Okay, so now talking about action potential, I think I can go a little bit fast because all of you are familiar with the concept of action potential. I was saying that this is from an experiment of mine when like 5 years ago, something like this. Yeah, when I was doing my postdoc in New York, and what I did here is that I record a single neuron like in an awake mice, like with like sharp electrode, and then like this is the trace of the neuron, like the action potential, there's a threshold computation of the neuron, and then in these experiments we were able to inject velocity in the neuron that we can find later in histology. So we have the histology of the neuron and the action potential of the neuron, right? This is cool, but these experiments are extremely painful to do, like very difficult to do, and we don't have much more than one neuron at much, or a couple of neurons, like in crazy experiments. But the truth is that like these action potentials, they are going to mobilize a lot of ions around the neuron, and this is something that we can like put in our advantage. If we put an electrode now outside of the neuron, but close to the neuron itself, we will be able to see some of these extracellular ions that the neuron is moving when it's generating its internal action potential. This is what you have here in a simulation. So this is from a colleague of mine, like 10 years ago, when he was doing his postdoc, I think in the same lab I did my postdoc. And then what you have here in the traces are like the different like changes in the extracellular like field potential. So basically what an extracellular recording channel will pick up around a neuron when this neuron is generating an action potential. And as you can see, this is a signal that we can see from outside. Again, because although we are used to see like the intracellular action potential, these intracellular action potentials, they mobilize a lot of ions like around. And then this is how our extracellular recordings actually look like. So what you see here is a compound of like the slow signal, the envelope, that this is the LFP, that we will talk a little bit later about the LFP, like it's like a combination of everything that is going on like in the circuit, and then the fast like action potential that are these small things, and these fast things like happening when single neurons are firing. Okay, as you see, like these extracellular action potentials, they go down, they don't go up, and the intracellular recordings, you see the action potential go up. I don't know if this is going to work, but this would be a question for you. So why intracellular action potential go up and extracellular action potentials go down? Like, any of you have like an idea about what this distinction? I will wait 15 seconds, really giving you some time to answer, because I know like online this creates some distance, and if no one says anything, I will tell the response. Okay, I see a lot of enthusiasm here. Okay, guys, like the response here is that like these action potentials happen because ions that are positive ions, like charged ions that are outside of the neuron, go inside the neuron, and these positive ions are outside, go inside, and this is why the neuron gets depolarized, right? Because positive neuron ions is getting inside the neuron, and with these positive ions, the membrane potential will go positive, and then these positive ions are being lost outside, and this loss of positive ions outside of the neuron in the extracellular space it means like a net decrease of charge in the exterior of the neuron, and this is why, like in effect, like the intracellular positive action potential is going to create a negative action potential. It's exactly outside of the neuron, right? So it's the specular version of the intracellular action potential. This that maybe seems like a trivial concept is something to bear in mind because we are going to use it later. Okay, like the unitary activity, the action potential is one of the most simple like electric brain signals that we can imagine. But as I was saying before, like with our electrodes, we also pick like this envelope. I have someone in the lobby. I have made right or whatever. Okay, so our electrodes were picking up these two type of signals, this action potential and then these slow envelopes. That this is what we call like the brain oscillations or the local field potential, right? So for studying and for talking about brain oscillations, we are going to talk all the time about a case of a study in which I did my PhD and half of my postdoc, and it's an event, a brain event that is very specific. That is something that we can distinguish in our recordings, and you will see it's quite important because it's an event, an electrical event, discrete in time, that has been directly related to memory consolidation. It could be like in a romantic fashion, that this like is kind of the noise of the brain when it's generating the brain memory consolidation. Is the artifact that the brain generates when it's like stabilizing memory traces. So this is like what these chart wave ripples are in the hippocampus, right? And like we like to study them because it's one way to link like electric signal and cognition, and it's something that like many labs have studied, and we have now a deep level of understanding. So I think it's an interesting case of study to illustrate what I am saying. Chart wave ripples are basically this is, I mean, it cannot be anything else than a signal, right? So it has basically this shape. So if we are recording, don't worry about the details because now it's not important. We have these electrodes inside the brain and we're taking electrical activity, and some electrical activity has this shape. Like in some channels, we see a high frequency component that you see here that we call this high frequency component ripple. Is high frequency because, as you can see, it moves very fast up and down in time, right? And then in some other channels, you see that around this high frequency event, we have a slower event that we call the chart wave. Don't worry again about the details. We are just introducing our case of a study that this particular electric activity, right? And that this is both like electric signal and it contains a brain oscillation. And this is something that we can record with our electrodes. Then these chart wave ripples, they are compounded by synaptic activity at some extent. And now we are going to talk a little bit more about the details. Like now I tell you that these ripples, this is something that we can record in the hippocampus. And it makes sense because I told you that this is the noise that the brain does when it's generating memory traces. And we know that memory, like some episodic memory, whatever, but like happens in the hippocampus. Like is like a important, a fundamental structure for generating memory, right? So then in this structure, when these ripples happen, like the hippocampus itself is generating memory. And for generating this, it needs inputs. And these inputs are going to be synaptic activity. And then is when I tell you a little bit more of information about like what the ripples are. So when ripples happen, the activity in CA3, I don't know how much you know about the hippocampus, but here we're going to talk about a very simple like circuit that is like several level of complexity, like more simple than what we really study, but it doesn't matter. So we are going to record most of the time like in CA1, that is one of the regions of the hippocampus, and this CA1 area received inputs from another area that is called CA3, right? And then I tell you that during the ripples, CA3 is projecting a lot of information to CA1, to these like pyramidal neurons in CA1. And these inputs go to the dendrites. And then these dendrites are gonna get the input from this CA3 as glutamate, right? That is released like in the synaptic cleft, right? So we are talking about like the synaptic component of the brain oscillation. And then like this glutamate is gonna open like sodium channels that are gonna go inside these pyramidal neurons. This is how the information is being sent from CA3 to CA1. And then what is happening? Like put it in context of what I was telling you before about the action potentials. So now, even though this is synaptic activity that it may not generate action potential, this is like the glutamate is opening like glutamate channel that are gonna be most of them like sodium channels. So sodium is gonna enter inside these neurons. And this is gonna create a lack of sodium channels in the extracellular space around the dendrites, the apical dendrites of this neuron. And this is going to create a negativity of the signal, of the brain signal in this case, around these dendrites. And this is what creates this charge wave. So the charge wave, that may be a signal, maybe not super fascinating when you see it like at first glance in your recordings, is super interesting because it's actually the loss of ions that is happening when CA3 is projecting information to CA1. And this is something that now that we know this, because we have this knowledge, we can compute and we can analyze and we can see how it changes across, like learning or whatever. And this is the kind of things like we do all the time. So brain oscillation, as you can see, are composed at some extent by synaptic activity, but they are also they're composed by action potential. And this is something that it look a little bit trivial because, okay, like you see some of these components here in the trace, right? Like obviously, like this action potential are gonna move a little bit the envelope, but even more, if many of these action potentials happen at the same time, then like this action potential will make a huge signal in the average envelope. And this is what is happening exactly in the ripple itself. So during the ripple, because these neurons are getting a lot of excitation from CA3, like the system in CA1, this local circuit is gonna get a surge of activation. And throughout local connectivity, this is gonna create a lot of like coactivity in the circuit. Is gonna create like some nonlinear like interaction between the neuron, and then many of these neurons are gonna fire at the same time. And when the neurons are firing at the same time, they are gonna sum each other and create kind of a populational action potential that we will be able to see in the local field potential. And this is what you see here, like in these throats. So each of these throats of the ripple, each of these like single like waves of the high frequency component of the ripple is actually a summation of a lot of pyramidal cells that are firing together with some level of coordination because they are getting a lot of excitation from CA3. And now we understand a little bit more, right? What is the ripple itself? Is this charge wave that is the synaptic activity entering into the light system of CA1, and then the superposition of a lot of action potential creating this high frequency component at the level of the ripple. It's a little bit more complicated, but I more or less like this is some like decent picture of what these events are. Then like the brain oscillations, they are composed also by some non-synaptic generator, for example, like intrinsic elements of the system, like gap junctions are a good example of things that can explain some of the variance of the local field potential, because basically like they are going to communicate neurons between them, right? Actually, like during ripples, it has been shown that like the gap junctions between pyramidal neurons will communicate, will tend to make the neurons to be coactive like more often, and this is gonna shape a little bit this population action potential that make the high frequency component of the ripple, right? So then we see how like an intrinsic property in this case of the neuron can shape the local field potential or the brain oscillations. And then something that is fundamental and we study and have to take into account all the time is the brain structure and the geometry of the region that we are studying. For example, we like to study the hippocampus a lot because the hippocampus like is a single layer with all the neurons next to each other with all the dendrites next to each other, and this is going to make like very beautiful brain signal. When we study the cortex, like for example, this is for a recording of mine where you have like CA1 recording and simultaneous like cortical recordings. When like in case of CA1, as I was saying, like it's a monolayer that is very well organized, and then the signal are beautiful. But if you start to overlap many of these processes that I tell you before, if now like one apical dendrite that is creating a beautiful signal, it coincides with the body of another neuron, whatever, like all this very complicated architecture that it happens in the cortex, then the brain signal become super, super boring. And this is what happened many times in the cortex, that many of these like interesting signals are being cancelled like by other processes, and then what we see is flatter and it's harder to study. So the brain geometry and architecture and the cellular architecture is actually fundamental in the brain signal that we are like studying. Then I'm gonna go a little bit fast with this because I don't think it's super important, and these are things that are quite obvious. Like there are different ways to study like brain oscillations. Like the classical ways was like electroencephalography, basically like putting electrodes like in the scalp, right? That this is not a very like how to say like high quality signal because has been like filtered out by the skin and it's very noisy, whatever. Then what we do most of the time is actually we put this system of electrodes inside the brain itself around the electrodes, and for us this is like the gold standard because here we can see like the local field potential and the spikes at the same time, and now like with the current technologies we can have like, for example, in this example I have like I think around 500 electrodes inside the brain. We can record more than several thousand of neurons like at the same time in like chronic implants while the animals are doing behavior and everything. And obviously like you can test, like you can have animals that are like model for diseases, you can try drugs, and you can do longitudinal experiments recording the brain activity. In other standards, people do like head fix preparation. So these are not chronic experiments. So here the animals are head fixed, and then you can open a cranial window and then lower the probe. And this is another standard with some advantages and some disadvantages, of course. Then with the analysis, I'm gonna go also a little bit fast because like the details can be a little bit tricky and we don't have time for this. And I'm gonna just tell us like the necessary so we can keep like moving forward. So as I was telling, like we have the raw data. In the raw data, it's gonna be like a composed signal with the LFP, with the brain oscillation, with the action potential. So we do some processing. So basically, we are gonna filter the signal only to get the spikes. So we can do a high pass filter, and this way only the very fastly happening things will survive, that are going to be mostly the spikes, and then we do some like analytical trick to distinguish the different neurons based on the fact that like the neurons, as you know, like they produce action potentials that are always the same, right? Like this is like it's not like an all or nothing like phenomenon. Like the amplitude and the shape of the action potential of a neuron is the same, and then if the electrode is stationary, the signal of the action potential of a given neuron is going to be the same all the recording long. And then we can use this in our advantage to be able to classify single neuron for our like raw recordings, right, using this analytical trick. So then we start with this raw data and we end up with local field potential, brain oscillation, and then a lot of neurons firing around this like brain rhythms with some behavior of the animal if the animal was like implanted like chronically, for example, and so on. And then a few things on the analysis again that I think will like help us to understand like some more complicated concepts later. Like because action potentials, we went from the raw signal to unitary activity now that were the action potential of individual neurons, and these are discrete events like action potentials that happen at some point during the recording, right, in some moment in time. And then we are going to study like the temporal relationship between this action potential. For example, let's say that we have two neurons, neuron A and neuron B, and each of the vertical lines here is an action potential from these two different neurons. And we can study the temporal relationship between these spikes, just like, for example, what you have here is a distribution of the distance between all the spikes of this neuron, meaning that I go from one spike that will be used as a reference, and then I compute the distance in time between this spike and all the rest. And then I do the same with this spike. And then I make an histogram of all these delays, and this is what you see here. And this is going to give us information about, on average, how much a neuron waits to fire one spike after it was firing, right? And this is important because, like, the principal neurons in the brain, they have very different temporal games, if you want to call it this way. For example, like pyramidal neurons are usually bursty neurons. Once they fire, they took a while to fire, but once they fire again very rapidly. And then we see this peak in this autocorrelogram. This is what these plots are. And interneurons are typically like regular firing neurons. They are firing all the time, and when they fire, they may or may not fire later. It really doesn't matter. They have like a more rapid relationship, and then they tend to have like these autocorrelograms. And then just like the shape of this autocorrelogram can help us to distinguish between interneurons and pyramidal cells, something that is like super important for our recording, because we are increasing like the kind of information we can extract for our recordings. Then another thing we can do is study the temporal relationship between neurons and things that are happening. For example, like the most simple thing, like following the scheme, would be to study the relationship between this neuron and this neuron. This is basically the same thing I was telling before, but now instead of comparing a neuron with itself, comparing a neuron with the other neuron. In this case, I take this spike as a reference, and then I like go throughout all the spikes of the other neuron, and I take all these delays and I make an histogram of them, and then I can keep going throughout all the spikes, like having all of them as reference, and then I make a distribution plot. And this distribution plot will tell me what is the temporal relationship between B and A, right? And in this case, if we see something like this, we know that the neurons, they have like common inputs, like the interpretation then is upon the experimenter, right? But they tend to co-fire. And this could be important for our experiment. For example, like if this changes upon a learning paradigm, we could interpret that these two neurons were related to the learning itself, right? But with the same approach, like for example, another like an interesting case of a study, like following this analysis, is like sometimes we find relationships that look like this. This is actually from a paper of mine like a couple of years ago. Like one neuron is firing, this is this kind of plot when we are studying the temporal relationship between these two neurons. And as you see here, when this neuron fires, then this other neuron is firing very rapidly and like in very reliably. So that means that these two neurons are connected. So this is a technology that allows us to see monosynaptic connections in behaving animals, something that obviously like is extremely powerful, right? And we could use exactly the same trick, and now not comparing like one neuron with other neuron. We could compare this neuron with another event, like for example, like something that is happening like in the environment, like something that the animal have to learn, or for example, the ripples. We could say, now I want to study like the relationship between neurons and ripples, and this is, for example, what they did in this study, and they saw how, like on average, during the ripples, the neurons were being active, right? So this is like these autocorrelograms and cross-correlograms is the analytic tool that allows us to study the temporal relationships between the neurons and another neurons and anything you could imagine. And now we are going to talk a little bit about, now with this we close the first part about the electric signal. I think that was the longest. Now we are going to talk about like manipulating this circuit, and then let's see if we have time to put the two things together like in the third part. So like the most simple way to manipulate the brain circuit is obviously with electricity, right? We are measuring electricity, and then we can also deliver electricity to the system and change it. And this is, for example, what people have been doing like for ages in the like LTP protocols, right? Like it was shown that if I guess that we all know what is an LTP protocol, right? Like long-term potentiation. Like basically these like I think like are 50 or 40 years old experiments when people show that like if you are recording this exact like a projection in the hippocampus I was telling you about, like CA3 towards like CA1, this connection is called like the Schaffer collateral. Like if you are recording a neuron here and you stimulate the Schaffer collateral, so in here you are basically stimulating the input to CA1 now in a slice. If you provide a high frequency perturbation, now this is how the circuit was being manipulated with a high frequency like stimulation, a high frequency electrical stimulation, then the connection between CA3 and CA1 get strengthened and get strengthened because like you are providing a lot of activation. Glutamate it's entering into the CA1 neuron and it's generating plasticity so that like the same event that was creating some signal before now produces twice the signal, right? I guess like we all more or less are familiar with at some extent the concept of LTP. And I mean these are like the experiments like doing electrical manipulation, but these have evolved a lot. Like this is, for example, like I like it a lot, like from it's an experiment from Michael Breck, and here they were doing manipulation not in a slice of a bulk of tissue. Here they were stimulating one single neuron, and they trained the animal to be able to detect this one single neuron activation. The task is a little bit complicated, but basically they trained the animal to be able to detect one single neuron stimulation and then leak like a port. And basically they showed that that was like possible, and it's an extreme case on how electrical stimulation are extreme, like super powerful, like to understand the brain. But electrical signals, in this case, they were creating a sort of plasticity. They were being perceived, but it's something that we can also use to perturb the system. For example, and now I go back a little bit to the ripples. I was telling you that like it was like hypothesized that ripples were related to memory consolidation, and then one of the most important like papers that was published on the matter was trying to do like a loss of function experiment. So if the ripples are related to memory consolidation, I don't think I have to like talk about them again, right? It's like the high frequency event with the charge wave, all of this, like CA3 projecting to CA1, whatever. So when these ripples are happening, like it was hypothesized that the neurons were like getting like interpreting associations like from CA3 to CA1, and it was important for memory consolidation. And then some researchers said, okay, then let's try to perturb it. If this is true, like if I perturb the ripples, in this case electrically, I should block at some extent like learning. And this is what they did in this experiment. So here you have an example of how like the perturbation of the ripple was working. So in the control condition, nothing. Like when a ripple was appearing, like they let the ripple happen, and then some millisecond later, they were giving some electrical stimulation into the brain to keep like the amount of electricity that was into the brain the same. And then in the real like experiment, what they were doing is that they were detecting ripples and very fast perturbing them. And this is the learning curve of the animals, in this case in a radial arm maze, but it doesn't matter. This has been replicated in many different tasks. And what you see here is that in the experiment, like itself, like in the red line, like the rats were taking longer to learn, and some of the rats will never like perform as the control one, meaning that like here, just blocking the ripple, like a very specific event that lasts like 50 milliseconds, you can block learning. And that was like one of the most important proofs that was published like to relate like memory consolidations and ripples. Another very cool and totally game changer like technology that we use to manipulate neural circuit is optogenetics. So the optogenetics are based in a very simple but clever principle. Like obviously we have neurons that are able to respond to light, because this is why you guys can see these slides, right? Like in the retina, we have some neurons that are light sensitive. So we can use this in our advantage. In this case, they were not using proteins from the retina, but from algae. And what the researchers did was to take some of these proteins and put them in neurons. This way, you have neurons that are going to be able to respond to light, but they are inside the brain, right? So no light is entering into the brain. But now this is something that you can use in your advantage, putting like optic fibers and then activating these neurons as if they were a switch, right? So this way you can interrogate neural circuits. And now what like was done later, and like our lab is doing, and the lab in which I was doing the postdoc was doing all the time, and they somehow like pioneer, was combining this neural activation with recordings. So you have manipulation and recording of the neurons at the same time. Even for example, just like to close like the case of study, the ripples, another, like I told you before about like a loss of function experiment that like this researcher, this French researcher, in actually in the lab in which I did postdoc in New York, like in the lab of Yuri Butsaki, published like some years ago. So what was missing here is a gain of function experiment, meaning like producing ripples and then seeing if the memory improves. And now, like that, we have this way to manipulate the circuit upon light, and that it was shown that actually now, like we can produce ripple, like kind of mimicking this input from CA3 that the neuron in C1 were experiencing. Like then they test like if these artificial ripples or these elongated ripples that were able to produce with light had an impact in memory consolidation. So before I told you that perturbing ripples, it kills memory. So if now we have a tool that allows us to produce ripples, that memory improves. And this is actually what they showed like a few years ago in the same lab that was published like the other papers, the lab in which I did the postdoc. So here now, this like the experiment itself that is again in red now goes like above the learning curve of the control animals, meaning that they were learning faster because they have more ripples. And then like in the last, I would say like the last commit, like the last iteration of this technology have been the generation of high resolution optogenetics, which like I mean are probes that combine like single like neuron, like body neuron size like LEDs and electrodes. So we can read out and read in like neural activity of circuits at real time. I'm gonna close like the manipulation part, talking about chemogenetics. That it goes a little bit off of what I said before, but I was told that also like this talk it's about drugs, about pharmacology, and this is like the angle of pharmacology of the manipulation I was telling you before. Maybe some of you are familiar, but basically, like chemogenetics are a very similar game of optogenetics, but here instead of using light to manipulate the neural circuits, we are going to use a ligand that is going to be an external ligand that is not able to change anything in the body unless we put a protein in some neurons using genetic tool that are sensitive like these proteins like to that ligand. This way, when we inject this ligand, and this is like the ligand is CNO, and this is the protein that is being used. So this protein is being expressed in some neurons that we want to study like the contribution to whatever, like to behavior or the function of the circuit of this neuron, whatever. Like once we inject the ligand, then like this, there are actually like excitatory and inhibitory flavors, but like this ligand will activate or inactivate only this population of neurons. So this is a very specific way also to manipulate this circuit. And I'm afraid that I don't have much time to go with this, like Claudia. Uh, I don't know if I can have like 5 minutes and go fast with important parts of this, or you want to close it here?
No. Go on.
Okay. So what we are really interested in the lab is how the brain represents information. And for doing this, we employ like the technologies and the tools I was telling you before in mice that are resolving memory tasks, basically. When we do this in the hippocampus, one of the things that is most striking is that we are going to find neurons mostly in CA1, this super interesting structure I was telling you before, because the ripples happen in the CA1 and so on. And these neurons are going to be able to represent the space. So these are the so-called like place cells. I guess that some of you are familiar with what place cells are. And this is what you see in this video. So this is a mouse going back and forth in a linear maze. So this is what you see in the trajectory, like the position of the mouse in different moments of the recording. I'm gonna go back so you can see this again. And then every dot is an action potential, in this case, from 5 different neurons. And as you can see, like this, at least these 4 colors, like pink, blue, green, and red, are neurons that are able to represent only a specific portion of the space, right? So these are place cells that are representing like that part of the space, such as the full combination of the 4 neurons is able to encapsulate the experience of this animal in this maze. The yellow neuron is also a pyramidal cell, but it's quite special because, as you can see, it seems to be not caring very much about this task, okay? But if I have time, like it will be handy later. In what we find in humans, for example. So how this is generalized to the case of humans, like a colleague of us here in the institute is Rodrigo Quiroga, and he found something that was hypothesized like many years before this paper, that neurons in the hippocampus, in case of humans, they are represented not only space, you know, more like multidimensional, like variables, like for example, like the social dimensions of the stimuli we are exposed to. Like the most celebrated case was the neuron of Jennifer Aniston. In this case, was a neuron that it was not in this case firing to a specific position of a maze. It was only firing to the concept of Jennifer Aniston and generalizing like her picture, like her name and everything, right? So we like to study these neurons in the hippocampus because we know that they are kind of representing portion of the world, and this is going to change when animals and humans are learning, right? So this is an entry point, a direct entry point to these high cognitive processes that we are very interested in. And then something like, to go again with the case of a study that is totally astonishing that happened during ripples, is that I told you that like these neurons were able to encapsulate the experience of the animals in the maze. And now we have the technologies to see how these neurons fire during ripples, because we can detect ripples, we can detect neurons, we can do these cross-correlograms, all these things I was telling you before. And now, when we put these things like together, here you see a ripple that now you are expert on this. You can distinguish a ripple like by the fly when you see a recording. You will see that the place cells that were representing the maze before, in many of these ripples, they are going to have the same exact structure that they have before. Meaning that, okay, yes, like I told you before that we have made like loss of functions and gain of function experiments that show that ripples are generating memory traces that are important for memory consolidation. But this is how the brain does it. So what it does during the ripple is that it replaced a fast forward version of previous experiencing. It's like it's studying what have seen before as a way to increase the synaptic strength between the elements that were important in experiences, such as the memory can be created, such as like the memory trace can be created, you know, across the relationship between these neurons. And then, Claudia, I don't want to go like, because here I can go on and on. For me, no problem. I enjoy this, but I don't know if this is going to be too much. Up to you. This is basically like I'm going to tell you just one, like a big sentence. So what we are interested in reality in the lab is why the neurons are firing here and why the neurons are firing here. So what are the real mechanisms that the brain has to produce this action potential? So what are the computation behind these spikes? And to do this is for what we combine this like optogenetics, high resolution optogenetics with recordings. So in very general terms, like we could, like a neuron cannot do more than fire when it gets excitation on when the inhibition that is getting is being removed. So we are able to extract this kind of information. I know that I am going a little bit fast because now I just want to close, but I can, if you guys can have time for question, then I will go to the details. But basically, this is information that we can extract with this technology. So like basically with the light pulses, we are able to sense the level of excitability of the neurons, and then we can see exactly what the neurons are doing. And like going to the end, I can tell you that we could resolve that when the neurons are representing concepts or when the neurons are representing the space, actually what is happening in the circuit is super interesting because like two things have to coincide. Like the neurons have to get excitation, but also inhibition has to be removed. So we have this match between these two independent strengths of computation that the neurons have to experience. So they finally fire and they are representing like a concept or a space or whatever they are representing. And I don't want to go more in the details, but it's interesting because in the Ripple is different and whatever, but like I don't want to extend myself. And this is basically what I have prepared for you guys. Sorry that maybe I was a little bit slow at the beginning and I didn't explain like the last part with enough depth, but I hope that more or less like the important concepts were clear.